Extopopala NEET Academy. Today we are going to learn the second chapter, biological classification. So guys, let's start with the NCERT. In this chapter, yesterday we have learned what all are important, what are the classification, types of classification, and today we will learn more about the classification. Okay, so let's start. So this classification have started very long before, and Aristotle was the first one to start this. So let's see how he did it. Aristotle was the earliest and he used symbol morphological characters. This can be an MCQ. Aristotle was the first one to do it and he used symbol morphological characters as the basis of classification. Okay, and now let's see. And because of using this symbol morphological characters, he classified the plants into trees, shrubs and herbs. Okay, and now in case of animals, he classified it into which have red blood cells and which doesn't have it. Okay, now it's clear. Aristotle was the first one to do it on the basis of morphological characters, and in plants, trees, shrubs and herbs, and in animals with and without the RBC. Okay, done. Now let's go into the next scientist who initiated it. The first one was Aristotle, the second one was Linnaeus. At the time of Linnaeus, he classified the organisms into two kingdoms. Okay, two kingdoms. As we studied yesterday, there are kingdom, phylum, class, order, and there are so many categories, right? And Aristotle classified all the organisms in the basis of their morphological characters, but Linnaeus classified into two kingdoms, right? Two kingdoms, and let's see what are the kingdoms. Kingdom Plantae and Kingdom Animaliae. And what were the drawbacks of this kingdom? This kingdom, two kingdom classification. The drawbacks were they did not distinguish between eukaryotes and prokaryotes. Okay, this was the first drawback. Then they didn't distinguish between unicellular and multicellular. Next one, photosynthetic and non-photosynthetic. So these were the drawbacks. Two kingdom classification drawbacks, they didn't distinguish between eukaryotes and prokaryotes. Then they didn't distinguish between unicellular and multicellular, and between photosynthesis and non-photosynthetic organisms. Okay, now let's see the larger number of and the great drawback of it was it didn't distinguish or it didn't include a large number of organisms into either of the category. There is Kingdom Plantae and there is Kingdom Animalia, but there are some organisms which didn't fit into both of these. So you should know about the drawbacks. It is an important question. Now let's see, besides gross morphology, other characteristics like cell structure, nature of wall, mode, habitat, all these have to be included. They got it that just because of morphological characters we cannot distinguish the organisms into groups. So there was a need of including all other characters. Now let's see, above of this two classification there were new classification. So let's see the newest and latest classifications. Okay, this table you don't have to study it now because we can use this table as a revision purpose. Okay. And now as per the NCERT, the latest one will be R.H. Whittaker classification. Not the latest one, but what you have to study is R.H. Whittaker classification. R.H. Whittaker classification is 5 kingdom classification. Okay, 5 kingdom classification, and it's the most important thing in this chapter. Let's see what are the kingdoms. You have to study all these 5 kingdom: Monera, Protista, Fungi, Plantae and Animalia. It's simple, Kingdom Plantae and Animalia. Okay, then Monera, Fungi, Protista. Okay, there are 5 kingdoms. Now let's see, when did R.H. Whittaker initiated or proposed the 5 kingdom classification? The year is 1969. This is an important MCQ and this should not be left behind. R.H. Whittaker 1969 and the 5 kingdom classification. And now comes the most important part here. It's the classification were based on? What were the basis of these classification done by R.H. Whittaker? So, let us see. There are 5 entities. There were 5 entities that R.H. Whittaker used to classify the organisms. What are those 5 entities? Cell structure, body organization, body organization and mode of nutrition, reproduction and phylogenetic characters. These 5 entities you should be clear with it. What are those? Cell structure, body organization, mode of reproduction, mode of nutrition and phylogenetic relationship. All these 5 entities should be clear, so that they can ask which one of the following doesn't include as the basis of R.H. Whittaker classification. So, you should be thorough about it. Repeat it again. Cell structure, body organization, mode of nutrition, mode of reproduction and phylogenetic relationship. Okay. Now, this was the 5 kingdom classification. After this 5 kingdom classification, they started a new classification where it had 3 domains. In the first chapter, we studied that the categories were till kingdom. Starting from the species, it were till kingdom. But a new classification system arose, and in which there were 3 domains. Okay, 3 domains. Domains means there are 3 domains. Okay. 3 domains, and the speciality of the domain is that among these 5 kingdom, only the Monera was divided into, the kingdom Monera was divided into 2 other groups. Kingdom Monera was differentiated into 2 other groups, and they are domain 1 and domain 2. And all the other 4 like Protista, fungi, Animalia and Plantae were belonged into the 3rd domain. So, you got it right. Monera was divided into 2 kingdoms. The Monera were divided into 2 domains, and all the other 4 included in the 3rd domain. Right. Now, let us see what are the 2 domains from the kingdom Monera. And the 2 domains were, okay, let us see. Thereby it became a 6 kingdom classification. Now you got right. There is 4 kingdoms, and there was already 5 kingdom, and in this 5 kingdom, except Monera, all the other belong to the same domain, and Monera remained divided into 2 domains. So, all total there was 6 kingdom, 2 in Monera, 2 kingdoms in Monera and 4 kingdoms the other rest. So, there arose a 6 kingdom classification. There is an order for the 6 kingdom classification, but it is not given in the NCERT, so it is not needed. Okay, let us see. 3 domain and divides the kingdom Monera into 2 domains, and leaving the remaining eukaryotic kingdom into 3rd domain, and thereby a 6 kingdom classification. Right. Now, let us see the difference between the 3 domains. The first domain consists of Monera, and the second domain again it is a part of Monera, and the 3rd domain all others, right. So, the first 2 domains were prokaryotes, and the 3rd domain were all the other 4 kingdoms except Monera they are eukaryotic. So, all the eukaryotic belonged into 3rd domain, and all the prokaryotes were divided into 2 domains. Thereby 6 kingdom classification. Clear? Now, let us see. Okay, and there, as we said, the linear system of classification had different drawbacks. Likewise, the R.H. Whittaker system also had some drawbacks, and the drawbacks are that they they placed prokaryotic bacteria with other groups which were eukaryotic. This was the drawback of earlier classification, and now this R.H. Whittaker made it change so that the mistake was revised. Okay. Okay. Now, let us see, the fungi had chitin. There were the earlier classification had the problem that all the plants were having cellulosic cell wall, and the fungi was having chitin. So, both the fungi and plants which were having different cell wall were placed in a same group. But now because of this R.S. Whittaker classification, they changed it into different groups. Okay. So this you have to note, the fungi had chitin and the green plants had a cellulosic cell wall. Fungi had chitin, green plants had cellulosic cell wall. Okay. Now all the prokaryotic organisms were grouped under kingdom Monera. As I said, all prokaryotic under kingdom Monera and all unicellular eukaryotes under kingdom Protista. So these are 2 MCQ points. All prokaryotic organism under Monera, all unicellular eukaryotic under Protista. Okay. And kingdom Protista has both together Chlamydomonas and Chlorella. This was a PYQ. Which group has Chlamydomonas and Chlorella? They asked this as a previous year question. Okay, and the answer was kingdom Protista. Done. Now let's see. Over time, an attempt has been made to evolve a classification system which was not just morphological, physiological and reproductive similarities, but it is also phylogenetic, that means based on the evolutionary relationship. As we know, the classification further developed. They were thinking that it should not be just the mode of reproduction, nutrition, just that. It should also be related to the phylogenetic characters. Now let's see what is phylogenetic characters. Phylogenetic relationship between the organism means the evolutionary relationship, how the organism came. As we all say that humans came from different pathways, right? Like as we say in the beginning that humans are related to monkeys, we say that, right? That is a kind of evolutionary relationship. So all the organisms have such type of evolutionary relationship, and this classification should also be based on evolutionary relationship. So you should not forget this word, phylogenetic relationship. Phylogenetic relationship. Okay. The word is phylogenetic. Morphological, physiological, reproductive similarities along with phylogenetic relationship. Okay. Now let's go into separate kingdoms. Today we will be learning about kingdom Monera, kingdom Protista, kingdom Fungi and kingdom Fungi. These 3 kingdom will be learning today, and kingdom Plantae and Animalia will be done later. Okay, let's go deep into the kingdom Monera. Kingdom Monera contains all the prokaryotic organisms, right? Bacteria are the sole members of kingdom Monera. Bacteria are the sole members of kingdom Monera. Then most abundant microorganism. They are the most abundant microorganisms. So 2 MCQs. Who are the sole members? Bacteria. Then most abundant microorganisms? Monera, bacteria. Okay. Now they also live in extreme habitat. This is a PYQ. Which organism live in extreme habitats? It's kingdom Monera. Okay. What are the extreme habitats? Hot spring, desert, snow and deep oceans. Done. Now bacteria are grouped under 4 categories. We divided the kingdom Monera, and in kingdom Monera the bacteria were divided into further 4 categories. Okay, 4 categories. Let's see, let's learn by this picture only. The first category was Bacilli. Bacilli means rod shaped. Then Spirilla. Spirilla means spiral shaped. And Vibrio. Vibrio means comma shaped. Okay, what is the next one? It's Coccus. Okay, so Coccus. Coccus is spherical, Bacilli is rod shaped, and Spirilla is spiral shaped, and Vibrio is comma shaped. And let's see this. They may ask from this picture. Even these pictures are important in NCERT. Let's see this. Bacilli is rod shaped. They may ask, Bacilli has spore. True or false? Bacilli has spore. Right? So it's true. Then they may ask, the Spirilla has flagellum. It's true or false? It's true, because Spirilla has the flagellum. Right? Now let's see. The bacterial structure is very simple because it's a tiny organism. The structure is very simple, but they are very complex in behavior. The properties are complex. Let's see what are the complex properties. They are the most extensive metabolic diversity. They have the most extensive metabolic diversity. Do you know how? There are many bacteria which make their own food. There are many bacteria which depend on others. They are bacteria which depend on chemicals, means chemoautotrophs. Such like this is what they meant by metabolic diversity. And there was a previous year question, I think it's in 2012 or something, which of the following kingdom shows the extensive nutritional biodiversity, nutritional diversity. Here, so you may confuse, extensive metabolic diversity is in kingdom Monera and extensive nutritional diversity is also in kingdom Monera only. Okay. And let's see, it can be autotrophic, it can be photosynthetic autotrophic, it can be chemosynthetic autotrophic, or it can be heterotrophs, right. And the main thing that you have to note here is that its majority of bacteria are heterotrophs, right. Now, as we said, the Monera were again divided into two domains. What are they? Archaebacteria and eubacteria. What are they? Repeat with me. Archaebacteria and eubacteria. You should not confuse between them. Okay. Archaebacteria are those which live in extreme habitats. Archaebacteria are those which live in extreme habitats. And as we said, we know the extreme habitats. It's salty areas, and they are, because salt is known as halo. Okay. Philes means loving. So halophiles, salt loving. Salty areas, halophiles. Then hot spring. Hot is thermoacidophiles. Philes means loving again. So hot springs, thermoacidophiles. And marshy areas, it's methanogen. M for M. Marshy areas, methanogen. So let's say again, salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. And how does this archaebacteria differ from other bacteria in having a different cell wall structure? Okay, this was also a PYQ. What is the difference between archaebacteria and eubacteria? So, what is the difference? The structure of cell wall is the difference. Okay. And because of this difference in their cell structure, the cell wall structure, they are able to live in extreme habitats. Okay, you just have to remember archaebacteria live in extreme habitat because of their cell wall structure. Don't forget this. Now, methanogen. More about methanogen. Methanogens are present in the gut of several ruminant animals such as cow and buffalo. Okay, the gut of ruminant animals are the marshy areas they mentioned, and these methanogens are present there. Just remember, in this gut they produce the methane. So, the name came methanogens, methane methanogens. They produce, responsible for the production of methane as a biogas from the dung of these animals. Now, let us go into the second group of Monera, it is eubacteria. There are thousands of eubacteria. Do you know what they meant by eu? Eu means true. So, eubacteria means true bacteria. True bacteria. There are thousands of other bacteria known as eubacteria. Okay. And they will be having a rigid cell wall. They will be having a rigid cell wall. If motile, they will be having a flagellum. If motile, they will be having a flagellum. And what did we said? Which of the four category? Which among the four category has a flagellum? Yes, spirillum has a flagellum. Okay. Now, the main guy of this eubacteria is cyanobacteria. Okay, cyanobacteria, which is also known as blue-green algae. Cyanobacteria also has a pigment known as chlorophyll A. Where do you find chlorophyll? Chlorophyll are seen in higher plants, right? Chlorophyll are seen in higher plants. So, they are, what are higher plants? Higher plants are photosynthetic autotrophs. Higher plants will do their metabolism from the photosynthesis. So, these cyanobacteria also does the photosynthetic autotroph procedure. Okay. And what are the characters of this cyanobacteria? They are unicellular, they are colonial or filamentous. They can be colonial. Colonial means they can be aggregated in colonies, or they can be filamentous like this. As this picture, they can be filamentous. Okay. Then they can be freshwater or marine or terrestrial algae. Okay. They can be unicellular, they can be colonial, they can be filamentous, freshwater, marine or terrestrial. All these points are important. Now these colonies are generally surrounded by gelatinous sheath. The colonies, as I said, these colonies or these filamentous is covered by a gelatinous sheath or known as mucilaginous sheath. Okay. And because of this, have you ever seen in water resources there will be some kind of bloom, and that bloom is caused by this bacteria. Okay, blooms in polluted water bodies. And this can also, some of the cells, means this is totally a cyanobacteria, and some of the cells, some particular cells can fix its own nitrogen from the environment. Some particular cells can fix its own nitrogen from the environment, and those cells are known as heterocyst. Okay, specialized cells. Those are the specialized cells which are able to produce nitrogen of their own. Okay, these organisms can fix atmospheric nitrogen in these cells. So this is the thing. Then now let's see the examples for this cyanobacteria. Examples for cyanobacteria are nostoc and anabina. Nostoc and anabina. And as they given here, they may ask, nostoc is a unicellular cyanobacteria. Is it right? No, because nostoc is a filamentous blue-green algae. Nostoc is a filamentous blue-green algae. You shouldn't skip these pictures and what written under these pictures, because it's an MCQ point. A filamentous blue-green algae, nostoc. Then coming into, as we said, there are different types of metabolism. Now let's go into chemosynthetic autotrophic bacteria, where it oxidizes various inorganic substances like nitrates, nitrites and ammonia, and use their energy for ATP production. So photosynthetic autotrophs we saw from by using chlorophyll A, and the example is cyanobacteria. And example for cyanobacteria are nostoc and anabina, and nostoc is a filamentous bacteria. Done, that portion is done. Now the most important thing there was heterocyst, the specialized cell for nitrogen fixing. Now in chemoautotrophs, they oxidize various inorganic substances to make energy, and with this energy they produce ATP. Okay. And what are the nutrients they use? Nitrogen, phosphorus, iron and sulfur. Done. Now we saw about the autotrophs. Now let's go into heterotrophic bacteria. We all know that if we keep milk, it will turn into curd. Okay, there is a procedure of making milk, making curd out of milk, and this is because of a bacteria known as lactobacillus. Okay, this bacteria produces curd out of milk, and this is a kind of heterotrophic bacteria. Got it? Heterotrophic bacteria. And they are decomposers. They decompose the milk into curd, right? This was also a PYQ. Which of the organism chain are decomposers? Are decomposers that help in turning milk into curd. So it was kingdom Monera only. Okay. And they are helpful from, as I said, curd from milk and production of antibiotic. This is also important. Antibiotic. Do you know what is meant by antibiotic? Antibiotic is drugs which are used against the bacterial infections. Okay. And fixing nitrogen in legume, as we studied in earlier classes, fixing nitrogen in legume plants. Okay. And some also cause diseases, and the organisms which cause disease are known as pathogen. Pathogen is the organisms that cause disease. Okay. It can be to human beings, crops, farm and animals. It's not so important, just remember it can be decomposers, it can be antibiotics, it can be used in fixing the nitrogen in legumes, and it can also be pathogens. Right. And what are the diseases caused by bacteria? What are the diseases caused by bacteria? Cholera, typhoid, tetanus, citrus canker. When I studied, I used to study it by like this, CT square, CT whole square. But now we know that it's easy to study all these, but CT square means cholera, citrus canker, tetanus and typhoid. Okay. These were the bacterial infections. Bacteria reproduce and now let us see about the reproduction. Bacteria reproduce mainly by fission. Fission is the process of their reproduction. Mainly, you should note the word, mainly by fission. And under unfavorable conditions, means there are favorable conditions for the organisms to reproduce, but under unfavorable condition means the condition where they can't reproduce, they have to produce spore. So basically here we will be discussing about 3 types of reproductive methods. First one was fission, next one production of spores under unfavorable condition, you have to note it, unfavorable condition, production of spore, and sexual reproduction by adopting a primitive DNA from one bacteria to the another. Okay. Then 3 types, fission, spore and adopting a primitive DNA from another DNA. Primitive DNA from one bacteria to another bacteria. Okay. And now comes the hero of the kingdom, Monera. Who is the hero of kingdom Monera? It's Mycoplasma. This was, many of the PYQ were based on this hero. It's Mycoplasma. Who is Mycoplasma? It is an organism which is smallest, which lack a cell wall. Mycoplasma is the smallest, it lack a cell wall and can survive without oxygen. So all these 3 points you have to note. What are the 3 points? It lacks a cell wall, it is the smallest and it can survive without oxygen. Done. And it is also pathogenic in humans and plants. 4 points, smallest, lacks cell wall, survive without oxygen and it is a pathogen. And now let us go into kingdom Protista. Kingdom Monera is over. What all you have to note in kingdom Monera? In kingdom Monera you have to know about the 4 categories, then you have to know about the the diversity, the metabolic diversity, then you have to know about archaebacteria, different archaebacteria, then eubacteria, cyanobacteria, example of cyanobacteria, then that heterocyst, then you have to know about chemosynthetic autotrophs, then heterotrophs and the Mycoplasma. These are the things that you have to note in Monera. Now kingdom Monera is over. Let us go into kingdom Protista. All single-celled eukaryotics are placed under kingdom Protista. And what are the examples for kingdom Protista? Chrysophytes, dinoflagellates, euglenoids, slime molds, protozoans. You shouldn't forget it, you have to repeat it again and again. Chrysophytes, dinoflagellates, slime molds and protozoans. Okay, euglenoids and protozoans. These things use it and read it again and again, remember and rewind it again and again, you won't forget these things. Okay. And what are the characteristics of Protista? It's primarily aquatic. Then it also forms a link with others like plants and animals. Dealing with plants and animals. Okay. It also forms a link with the higher plants and animals. Then it has a nucleus. It has a well-defined nucleus. Did the Monera had a well-defined nucleus? No. So this have a well-defined nucleus and they have membrane-bound organelles. These are the important points. They are primarily aquatic. They have nucleus, well-defined nucleus and membrane-bound organelles. And they have flagellae. In kingdom Monera, some of them had flagellae, but here they have flagellae or cilia. They have some sort of locomotory agents, means the type of cells that helps them to move. Okay, flagella or cilia, and they produce asexually and sexually, both asexually and sexually. Done. Now let us go deep into the groups. Chrysophytes. Chrysophytes include diatoms and golden algae. Golden algae is also known as desmids. This is again a PYQ. Diatoms, golden algae and desmids. Golden algae is also known as desmids. Okay. Now let us see the next point. It's freshwater as well as in marine environment. This is seen in, as we said, it's primarily aquatic, but here it's freshwater or marine. Okay. And they are microscopic and float passively in water current, also known as plankton. This statement was itself a PYQ. Microscopic floats passively on water. They can ask floats actively on water, but it's not float actively, it's float passively on the water. Okay, and this is the most important thing in protesta. Diatoms has a cell wall. These diatoms has the cell wall, and this cell wall is made up of two thin overlapping cells, two thin overlapping shells, and they fit together to form a soap box. See, this is a diatom, this is a diatom, and this diatom have a particular cell wall. This diatom have a particular cell wall. This is the cell wall and this is the cell wall, and these two cell walls fit together like a soap box, and this cell wall fit together like a soap box, and this cell wall is made of silica. Cell wall is made of silica. Okay, it's overlapping shells embedded with silica, and because it is made up of silica, they are so strong, it's difficult to destruct. Okay, and because it is difficult to destruct, it is after the death of the organism. The organism is inside the shell, right? Organism is inside the shell, but the organism dies, but the shell remains intact, and in the water bodies the cell remains there for millions and millions of years, and this stored as such in the depth of water, and they are known as the diatomaceous earth. Let's see, diatomaceous earth. The accumulation over billions of years of these silica shells are known as diatomaceous earth. Okay, the cell walls are indestructible, it fits in a soap box, then okay, and this will be gritty because of the silica content, it will be gritty. Okay, and because of this it can be used in polishing, filtration of oils and syrups. So the uses also should be noted. And the most important point, they are the chief producers of oceans. Who is the chief producers of oceans? Diatoms. Okay. Coming into the next group, dinoflagellates. These are mostly marine. Chrysophytes were freshwater or marine water. Freshwater living or marine living. This is mostly marine and is photosynthetic. Okay, they are colored. Dinoflagellates are colored. They can be yellow, green, brown, blue, red. Okay, and what determines the color? The pigment present on their cell wall determines the color. Okay, and what is present in their cell wall? In chrysophytes, the cell wall was having silica, but dinoflagellates have a cell wall with stiff cellulose plates. And dinoflagellates, see the name, dinoflagellates means di in number of flagellates, two flagellates, two flagellas. Okay, two flagellas. Dinoflagellates have two flagella. Done. And one is longitudinally. See the flagella? One is longitudinally and the other flagella is through the furrow between the organism. There is a furrow. Furrow means there is a cave-like structure. It is a part of the organism, and one filament is or one flagella is longitudinally placed and the second flagella is placed in the furrow. Okay. Two flagella longitudinally and other transversely in the furrow between the wall plates. And the example for dinoflagellates is red dinoflagellate, also known as goniolax. And you know what, these dinoflagellates when get accumulated, they will be so much in water and they will form a red tide. They have color, as I said, because of this pigment they will feel like this ocean is red. So it is known as red tide. See up here, red. Okay, and this is toxic. So toxins released can kill marine animals such as fishes. Now the next group is euglenoids. Euglenoids are majority of the freshwater organisms. The dinoflagellates was mostly marine, but eugleno was freshwater. Euglenoids, E and F for flagella, F for the freshwater. Just write these like in your NCERT so that it will be easier next time when you see. Okay, euglenoids, E, so F for the freshwater, they are mostly freshwater. Then found in the freshwater organisms found in the stagnant water. Okay, freshwater organisms found in the stagnant water instead of cell wall. As we said, all of them have cell wall and different things on their cell wall. Here on the cell wall, what does they have? A pellicle. It's a protein rich layer. Protein rich layer known as pellicle is the presence of their cell wall. There is a presence of protein rich layer called pellicle, and because of this the euglena can be flexible. Euglena is usually flexible in their path. See the picture? Don't you feel that it's flexible? It's just because of the presence of their cell wall, which is a pellicle which is protein rich. Okay, and this also have two flagella. See, two flagella. One flagella it's this, and the second flagella is this. Means one is long, one is short. There are two flagella. Okay, done. Now let's see two flagella, short and long. Then photosynthetic. There is a characteristic of this organism, special characteristic. They are photosynthetic in presence of sunlight. Deprived of sunlight, they behave like heterotrophs. In sunlight, they are autotrophs. When there is no sunlight, it's heterotrophs. Okay. Now the pigments of euglenoids are identical to those present in the higher plants. As we said in the beginning, these protista links the higher plants. There are links with the higher plants. So this euglenoid have pigments which are similar to the higher plants. And example for euglenoids is easy euglena. Okay, now going into slime molds. Slime molds are saprophytic protist. Okay, these saprophytic protist, saprophytic means dead animals. They are seen along the decaying twigs and leaves and engulfing the organic material. Okay, they will engulf, they will make the organic material inside them. Okay, and in suitable conditions, they forms aggregate called plasmodium, and which may grow and spread over several feet. Okay, and during unfavorable conditions, they can form. During unfavorable conditions, the plasmoid differentiates and forms fruiting bodies. Okay, these two things you have to note. What is the things that you have to note? During favorable conditions, they form plasmodium. And during unfavorable conditions, they will differentiate. These during favorable conditions, see, during favorable condition, they will form a plasmodium. And they can differentiate. During unfavorable conditions, they will form spores. They can differentiate into fruiting bodies. This is the differentiation, and they can form fruiting bodies. This is the fruiting body, and each of them can be a spore. Each of them will be having a spore at the end. See this. During favorable conditions, they can form plasmodium and they can grow. The plasmodium will grow. Then unfavorable condition, plasmodium differentiate and forms fruiting bodies, and they will be having the spore at the tips. Just remember the point, spore at the tips of fruiting body. And the spore possesses true walls. Okay, they are extremely resistant and survive for many years. As we said, the diatomaceous earth which are resistant and which are resistant to destruction and which will be there for all the years, right? So the spores also will be extremely resistant and will be preserved for many years. Okay, the spores are dispersed by air currents. Most of the things that I underline are important. Okay, now coming into protozoans. Protozoans are heterotrophs. And they live as predators or parasites. They can be either predator or parasite, and they have primitive relationship with animals. Who are having relationship with plants? Euglenoids. And who was having relationship with animals? It's protozoans. Okay, in protozoan there are 4 types. First one is amoeboid protozoans. They live in fresh water, sea water and moist soil. Okay, just read along with me, it will be easy for you. And then this just remember amoeboid protozoan is having pseudopodia. Okay, pseudopodia means false feet. And this amoeboid protozoan also has silica shell. Where did we see the silica shell before? It was in chrysophytes. Okay, chrysophytes. In diatoms we have the silica shells, again in amoeboid protozoan silica shells. Okay, and endamoeba is the example and it acts as parasite. Okay, now flagellated protozoan, second group. It have flagella, as the name suggests it have flagella. It can be parasitic or free living. It can be free living or parasitic. Okay, the parasitic form causes disease such as sleeping sickness. Its example is trypanosoma. Sleeping sickness is the disease caused and its example is trypanosoma. Okay. Ciliated protozoans, these are aquatic actively moving organisms. Ciliated protozoans. Ciliated protozoans are aquatic actively moving organism because of the presence of thousands of cilia. Okay. We said in the beginning they may be having either flagella or cilia. So, ciliated protozoans having cilia and they have a cavity known as gullet. They have a cavity known as gullet. Okay. This gullet opens to the outside of the cell surface. Okay. There is a cavity known as gullet and into this cavity all the food particles come inside so that it can feed. Okay. And this helps in the movement of rows of cilia. There are so many cilia. Just see this. Ciliated protozoan may look like this. They have so many cilia and because of the movement of all of these cilia, they can move. Okay. And this will help them to move the food particles into the gullet. Gullet means into the cavity inside their body. And the example is paramecium. You should be clear with the examples. For flagellated, the example of disease is trypanosoma. For protozoan, for amoeboid protozoan, it's endamoeba. For ciliated, it's paramecium. And in sporozoans, as the name suggests, it produces spores. Diverse organisms that have infectious spore-like stage in their life cycle. Okay. Plasmodium, malarial parasite which cause malaria. Okay. And this malaria is caused by plasmodium, that only you have to note. Now coming into the last kingdom that we are going to study today, it's kingdom fungi. Okay. Fungi constitute a unique kingdom of heterotrophic organism. This was a PYQ. Fungi belongs to heterotrophic organism. Fungi is a heterotrophic organism. Okay. Now the common mushroom you eat, the mushroom that you eat, this is basic. Mushroom you eat, toadstools are fungi. Toadstools are, or they can ask like this, toadstools belongs to fungi. Okay. Now the white spots on mustard leaves are also parasitic fungi. Then it's parasitic fungi, don't forget that. White spots on mustard leaves, parasitic fungi. Then the only unicellular fungi, this is most important thing, only unicellular fungi is yeast. Okay. These all they have described the examples. Wheat rust causing Puccinia, rust Puccinia, okay. Rust Puccinia. Then antibiotic source is penicillium, rust Puccinia, antibiotic penicillium. Then fungi are cosmopolitan. Cosmopolitan means seen everywhere in normal places, in decaying, in dung, everywhere it's seen. So it's cosmopolitan. It's in air, water, and soil, and animals and plants. Okay. They prefer to grow in warm and humid condition. Warm and humid, as we say that fungi will say that food will be spoiled if they are kept in warm and humid condition because of the fungi. Okay. Fungi are filamentous. Fungi are mostly filamentous. Only yeast is unicellular, so it can't be filamentous. Mostly other fungi are filamentous. Okay. Their bodies, this filamentous fungus will be having their bodies will be having long, slender, thread-like structures called hyphae. Every fungus will be having, most of the fungus will be having long thread-like structure known as hyphae. And when we take the section of this hyphae, the hyphae may look like this. Okay. The hyphae may look like this. And the network of hyphae, this is just one hyphae. The network of hyphae means many hyphaes. The network of hyphae is known as mycelium. Network of hyphae is known as mycelium. And there is another property. The hyphae can be multinucleated. If they are multinucleated with continuous continuous multinucleated cytoplasm, then they are known as cenocytic hyphae. They are known as cenocytic hyphae. So, this is cenocytic hyphae. But if they are divided, septate, branched, if they are branched or if they are septate, then they are not cenocytic hyphae. Okay. The septae means the branching, okay, branching or the division between the tubes. Okay. The cell walls of fungi are composed of chitin and polysaccharide, as we said in the beginning. Cell wall is chitin and polysaccharides. Now, the most of fungi are heterotrophic and soluble, or they take the soluble organic substance and from dead substance. From dead substance, then they are known as saprophytes. Okay, dead saprophytes. And if they are living on plants and animals, then they are known as parasites. This we studied in earlier classes. Okay. In dead, it is known as saprophytes. In if they are on living plants and animals, then they are known as parasites. And there is another category, symbionts. Symbionts means those which are living on the if they are having a combination, if they are paired with algae, if the fungi is paired with algae, it is known as lichen, and if the fungi is paired with the root of higher plants, it is known as mycorrhiza. Okay, these two things are PYQs again. If the symbionts, okay, they have given us two examples for symbionts. What is symbionts? Symbionts are mutual relationship. Okay, mutual relationship where both have the benefits. So, lichen, what is lichen? Lichen is algae plus fungi. Then what was the next one? Mycorrhiza. Mycorrhiza is fungi plus root of higher plants. Okay, reproduction in fungi can take place in vegetative method. Vegetative means fragmentation, fission and budding. All these are vegetative. Okay. Now, what asexual reproduction by spores can also be taken place. Vegetative method means fragmentation, fission, budding. Okay. There is no spores, but in asexual reproduction there is spores. Asexual reproduction, spores. Sexual reproduction, again there is spores. Okay. And what are the asexual spores? Conidia, sporangiospores and zoospores. So, you should not confuse between the asexual spores and sexual spores. Asexual spores are conidia, sporangiospore and zoospore. Okay. Sexual spores are oospores, ascospores and basidiospore. As just now itself remember that ascospore for ascomycetes and basidiospores for basidiomycetes. Okay. And the various spores are produced in distinct structures called fruiting bodies. Do you remember that while studying slime molds we said that there are fruiting bodies, and from the fruiting bodies comes the spore. Okay. So, here it applies again, produced in distinct structures called fruiting bodies. These spores are produced in distinct structures called fruiting bodies. Okay. Now, let us see, this was a PYQ again. Protoplasm, the fusion of protoplasm is known as plasmogamy between two motile or non-motile gametes. Okay. Fusion of two nuclei is known as karyogamy. Meiosis in the zygote results in haploid spores. Do you know how it is done? This is a zygote. Zygote will be having zygote will be diploid. Okay. So, in the meiosis is a reduction division where the number of nucleus will be decreased, it will be haploid. From the diploid it will produce 4 haploid spores. From the diploid it will be producing 4 haploid spores. We will study later about the in detail about the meiosis. Okay. Just remember this, meiosis in zygote will result in haploid spores. Okay, because it is a reduction division. When fungus reproduce sexually, 2 haploid hyphae of compatible means 2 haploid hyphae will be coming together and mating. They will fuse. Just see this. These are 2 spores. One haploid spore, the second haploid spore. They will come and fuse. They will come and fuse to form a 2N structure. But in some type of fungi, they will immediately form this 2N structure. But in some other types, some different type of fungi, they won't form this 2N. Instead of that, they will belong in this N plus N position. Okay, N plus N. That phase is known as dikaryon phase. That phase is known as dikaryon phase. That phase is known as dikaryon phase or dikaryophase. Okay, and what are the examples of organisms, or what are the example of types of fungi which belongs to the dikaryophase? They are ascomycetes and basidiomycetes. Ascomycetes and basidiomycetes. Okay. Now later there will be an n plus n, but later they will fuse to form 2n stage, and this 2n zygote will be later divided by meiosis to form haploid spores. The spores are always haploid. Spores are always haploid. This is important. Spores are always haploid. Okay. Now on the basis of the morphology of this hyphae and the type of spore, type of reproduction, this fungi is again divided into 4 groups. Okay. They are, the first group is phycomycetes. Phycomycetes is found in aquatic habitats. Okay. Again in decaying wood, moist and damp places. It's common property of fungi. Then can also be parasite on plants. Okay. Mycelium is aseptate. This is important. Okay. Mycelium is aseptate. Mycelium is aseptate. Mycelium is aseptate. Means it's cenocytic. It will be continuous multinucleated structure, cenocytic, aseptate or cenocytic. Now their asexual reproduction by the zoospore. Zoospore is motile. Just remember that zoospore is motile, and by aplanospore. Aplanospore is non-motile. Zoospore motile and aplanospore non-motile. Okay. And these spores are endogenously produced in sporangium. Okay. These are the points that you have to note. I will mark it again. Okay. I will mark it clear so that you just have to remember only these points. The first point is that the mycelium is aseptate. The second point is that the asexual reproduction by zoospore, which is motile, aplanospore, which is non-motile. And the third point is that it is endogenously produced in the sporangium. Okay, endogenously means inside. Endogenously means inside, it is produced inside. Exogenously means it is produced outside. Exogenously, this is endogenously. This is exogenously. Exo means outside, endo inside. Okay. And a zygospore is formed by the fusion of 2 gametes. It's the normal thing only. And if both of the spores which are mating are of same morphology, then it is known as isogamous. Iso means same, similar. If it is of different, it's dissimilar, then it's anisogamous or oogamous. Okay, now what are the examples? Mucor, Rhizopus, Albugo. Mucor, Rhizopus and Albugo. Rhizopus is the bread mold. Rhizopus is the bread mold and Albugo is the parasitic fungi on mustard. As we said in the beginning, white spot on mustard, Albugo. Okay. Now just see here. This one, say whether it is exospore or endospore. It's in the outside, right? Spore is in the outside, so it's exospore, exogenously produced. Okay. Now let's go into the next group. It's ascomycetes. Ascomycetes is also commonly known as sac fungi. Ascomycetes is commonly known as sac fungi. Phycomycetes is commonly known as bracket fungi, but ascomycetes is commonly known as sac fungi. Ascomycetes are mostly multicellular, but there is only one unicellular fungi, right? The unicellular fungi belongs to ascomycetes, and that is our yeast. Yeast is also known as Saccharomyces. Yeast is also known as Saccharomyces. And Penicillium is also belong to the ascomycetes group. They are saprophytic as the basis, saprophytic, decomposes, parasitic, coprophilous. And this coprophilous means growing on the dung. Okay. Now their mycelium. This comes the important point. Mycelium is branched and septate. The phycomycetes are aseptate, means it is not branched and it is aseptate, multinucleated. Here it will be uninucleated. See, in this mycelium, if it is septate, if there is septas in between, this is known as septa. Okay, if there is septa in between, it will be uninucleated. But if there is no septa, it will be multinucleated. Okay. Now the asexual spores. Asexual spores are conidia. There are different asexual spores, right? There the asexual spores in phycomycetes, the asexual spore was zoospores and aplanospores. In ascomycetes, it's conidia, and it is produced exogenously, and this mycelium is called as conidiophores. So just these points: asexual spores, conidia exogenously produced on conidiophores. Okay. And sexual spores are called ascospores, we said in the beginning, ascospores for ascomycetes. Ascospores which are produced endogenously in sac-like structures called ascae, singular ascae structures. Okay. In ascae structures there is production of this ascospores. Okay. And what are the examples? And fruiting body, the name of fruiting body is ascocarp. Examples are Aspergillus, claviceps and Neurospora, and Neurospora is used in biochemical and genetic work. Okay. And morels or truffles are also belonging to this category only. And now see the difference between asexual and sexual spores of ascomycetes. Asexual spores, see asexual spores is conidia. The asexual spore is conidia, it is exogenously produced and is produced on conidiophores. Okay, produced on conidiophores. In sexual, it's ascospore. It is produced endogenously. And it's produced inside the ascae. And the fruiting body is ascocarp. Done. Now basidiomycetes. Commonly known forms of basidiomycetes are mushrooms. The mushrooms that we eat. Okay, bracket fungi or puffballs. Okay. And they grow in soil, logs, tree stems. It's the basis only. And plant bodies are, and they also grow in plant body as parasites. This is example, rust and smut. So rust and smut. See this, USPR. This is a mnemonic, USPR. US means smut by Astilago. PR means Puccinia causes rust. USPR, Astilago smut, Puccinia rust. Okay. Rust and smut. The mycelium is branched and septate. Here comes the important point. Branched and septa means it has septa. Okay. Then asexual spores are not found. They may ask like this. Basidiospores are the asexual spores of basidiomycetes. Is it right? No, because they are not found. Asexual spores are not found. But vegetative reproduction like fragmentation is common. Now sex organs are absent. Okay. But plasmogamy is brought about by fusion of two gametes. Okay. Plasmogamy is present but sexual reproduction is absent. And they will result in dikaryotic structure. As we said in the beginning, there are some type of fungi which will form this dikaryon phase, ascomycetes and basidiomycetes. Okay, dikaryotic, and which will give rise to basidium. Okay, in ascae, as we said ascae, here it is basidium. And karyogamy, after formation of this fusion of plasma cells, after the fusion of plasma, then they will fuse the nucleus. It is known as karyogamy, and after that meiosis takes place and spore forms. It is simple procedure only. First it will plasmogamy will happen, then karyogamy will happen. Karyogamy means the fusion of nucleus. Then what happens? The meiosis, so production of 4 haploid spores. Okay. And see this, how is the basidiospores? Here the basidiospores is sexual spores. Okay, exogenously produced on the basidium. And now what are the examples? And the fruiting body is basidiocarp. And examples are Agaricus, Astilago, Puccinia. Agaricus, Astilago, and Puccinia. As I said, just remember this USPR. It's Astilago smut, Puccinia rust. Okay. Deuteromites is the last group of this fungi. Okay. It is imperfect fungi. It is also known as imperfect fungi. Astilago is also this Deuteromites is also known as imperfect fungi. Okay, imperfect fungi. It has only, why it is known as imperfect? It doesn't have a sexual phase. So it is known as imperfect fungi. Only asexual or vegetative phase of the fungi are known. Only the asexual or vegetative phase of the fungi are known. When the sexual phase are known, See, in the group of fungi, it's only asexual phase. If the sexual phase is known, it will be transferred into another groups like Phyco, Ascor, Basidio. Okay, so only asexual phase is known for the deuteromites. Okay, deuteromites reproduce. They have asexual phase, so asexual spores, and their asexual spore is known as conidia. Their asexual spore is known as conidia. Mycelium is septate and branched. Mycelium is septate and branched. Okay, some are saprophytes and or parasites. Some of them are saprophytes or parasites, but a large number of them are decomposers of litter and help in mineral recycling. Okay, large numbers of them are decomposers of litter and helps in mineral recycling. It's a PYQ. Okay. What are the examples? Examples are Alternaria, Colatotrichum, Trichodoma. I remember that when I used to study this, I was singing this song like Alternaria, Trichodoma, Colatotrichum. Alternaria, Trichodoma, Colatotrichum. Okay. Now it's just introduction about the kingdom Plantae and kingdom Animalia. Let's see. Just take 2 minutes break. Okay. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organism. All eukaryotic chlorophyll containing organism. We studied a prokaryotic organism having chlorophyll. Who was that? Cyanobacteria. Okay. Some of them will be partially heterotrophic, like insectivorous plants. They are heterotrophic, like dependent on other things. So example is Venus flytrap and insectivorous plant, and Venus flytrap and bladderwort. Okay, and this cascata is a parasite, plant parasite. Okay, then the cell wall is mainly made of cellulose. This you will study later, so it's fine. Two distinct phases are the diploid sporophytic and haploid gametophytic, but you will learn about it later. And alternation of generation are there in plants. Okay, alternation of generation means between the diploid and haploid phases. There will be alternation between diploid and haploid phases. Okay, now kingdom Animalia, they are heterotrophic eukaryotic. They were autotrophic eukaryotic plants. Here it is, animals is heterotrophic eukaryotic. Okay, they are multicellular and lack their cell wall. They don't have cell wall. Animals doesn't have a cell wall. They digest their food in internal cavity, as we know, we humans. Okay, and the process is known as holozoic ingestion of the food. Okay, sexual reproduction is by copulation of male and female, followed by embryological development. Now comes the important thing: viroids, virus, and prions and lichens. Virus doesn't belong to any of the classification, so they were kept aside as a different one. Okay, virus, viroid, and prions. Viruses are non-cellular organisms and characterized by having an inert crystalline structure outside the living cell. They are having a inert crystalline structure. They have inert crystalline structure outside the living cell. Okay. Just see this picture. It's tobacco mosaic virus. Tobacco mosaic virus. Tobacco mosaic virus. It has an RNA, means genetic material. Then it is covered by capsid, which is the proteinaceous structure. Okay. They will infect the cells and take over their machinery of the just in virus, there is a virus. Okay, this virus infect a host cell. This virus infect a host cell, and they will take over the machinery of the host cell and they will spread inside the body. This is how they work. Virus get inside the host cell and they will take all the control of host and spread. Okay, this is how they work. And virus means venom or poison. Okay. Now these things that I have marked are important. Ivanovsky recognized these organisms as the cause of mosaic disease of tobacco. Okay. Then the next one was they were smaller than bacteria because they passed through the bacteria proof filters. Do you know what it meant by this? Bacteria proof filters means the bacteria will be remained in these filters, and even this in this filters also, even by using these bacteria proof filters, virus was gone down. Okay, so they passed through the bacteria proof filters. So viruses are smaller than these bacteria. Okay, Then MW Bejernak. These are the important things. Okay, MW Bejernak demonstrated the extract of the infected plant. Means there is an infected tobacco plant, and the infected tobacco plant, if we take the extract, it will infect another healthy plant. Then that fluid was known as Contagium viva fluidum. Then W.M. Stanley, he showed the virus can be crystallized or crystals containing largely of proteins. Okay. And viruses are obligate parasites. In addition to proteins, they also have genetic material, and they could be either RNA or DNA, and there is a PYQ, which is the infectious structure of virus. It's genetic material or RNA, it's not the protein. Okay. No virus contain both RNA and DNA. No virus has both RNA and DNA. Then a virus is a nuclear protein and the genetic material is the infectious. Okay. The virus that infect the plants have single stranded RNA, and virus that infect the animals have either single or double stranded RNA or double stranded DNA. Okay. Plants single stranded RNA, and in animals either single or double stranded RNA or double stranded DNA. Okay. And the viruses that are affecting the bacteria are known as bacterial viruses or bacteriophages. Okay. They have double stranded DNA. And capsid is made of small subunit known as capsomeres. So this totally is known as capsid, and each of them is known as capsomeres. These capsomeres can be arranged in helical or polyhedral or geometric forms. This was a PYQ. All the virus have helical capsomeres. Is it right? No, they can be arranged in helical or polyhedral or geometric forms. See here. Here it is arranged in a geometric form. Okay. Now, what are the diseases caused by virus? Mumps, smallpox, herpes, influenza, AIDS. And in plants, it's mosaic formation, leaf rolling, curling, yellowing, vein clearing, dwarfing, and stunted growth. Okay. These things you have to note and study specifically. Now, T.O. Dehner, this was again a PYQ. T.O. Dehner, who found out the organisms known as viroids. Who was that? T.O. Dehner. Okay. These viroids are smaller than virus. Virus itself is small, and viroids is smaller than viruses. And what is the difference between virus and viroids? Virus has both genetic material and protein coat. Okay. But the viroids doesn't have a protein coat. That is the difference. Viroids doesn't have a protein coat, and the genetic material will be RNA, and that will be of low molecular weight. See, smaller than RNA will be it was found to be a free RNA, which means lack the protein coat, and it named viroid, and low molecular weight RNA. Okay. And prions are folded organisms. Prions are infectious. Prions causes infectious neurological disease. Neurological disease means which affects the nervous system. Okay. And they were abnormally folded organisms. What are prions? Prions are abnormally folded organisms, which we will study further in our higher studies. We will study more about it. Just now you have to note that they are abnormally folded proteins, and the agent was similar to size in virus. What is this size? It is similar to the virus. And what does it cause? Bovine spongiform encephalopathy, and it is also known as mad cow disease and Sear-Jacobs disease. Okay. Lichens, as we said before, it is a symbiotic association between algae and fungi. Okay. The algal component is known as phycobond, and the fungal component is known as mycobiont. Okay. And the algae will prepare food for fungi, and the fungi will provide the shelter and absorb nutrients and water. So they are both mutually useful. Algae will give the food, fungi will give the shelter. Okay. And they are very good pollution indicators, which means wherever there is lichens are present, the place will be free of pollution. Okay. Means these lichens grow at most pollution free areas only. So they are very good pollution indicators. Do not grow in polluted areas. Done. So the chapter is done. Now let's go into the previous year questions. Okay. There are almost 20 questions. We will do it first. Okay. One of the major component of cell wall of most of the fungi. What is the cell wall? Component of cell wall in fungi is chitin. Okay. Let's see the next one. Nuclear membrane is absent. Nuclear membrane is absent in nostoc, because nostoc is the Monera. Monera doesn't have a nuclear membrane. The table that I said you have to use for revision purposes. This table I didn't mention about it, but you have to use this table for your revision, solely for your revision, because everything is there in this table only. So you can use this table for the revision purposes, and in this table it's written the characteristics. Okay. So nuclear membrane is absent in Nostoc. Next one, viroids differ from virus. As I said, they are RNA molecules with or without protein coat? Without protein coat. Okay. Next one, archaebacteria differ from eubacteria. This I said, it's the cell membrane structure. Okay, the structure of their cell membrane or cell wall, the structure of their cell wall. Okay. Now match the column. Saprophyte means decomposition of dead saprophytic. Okay, parasite is living on living plants or animals. Lichens is symbiotic association with fungi. No, it's symbiotic association with algae and fungi. Mycorrhiza on roots. Okay, so A to B3. Where is A to B3? This is the right answer. Okay. Next one, choose the wrong statement. Yeast is unicellular and useful in fermentation. It's right. Neurospora used in study of biochemical genetics. Right. Morel and truffles are poisonous. Morels and truffles are poisonous. Is it poisonous? No, they can be used in food. Okay, morels and truffles. Next, ciliates differ from all other protozoans. Ciliates will be having cilia. Okay, and having a contractile, it will be having two contractile vacuole, having two type of nuclei, using flagella for locomotion. Does it use flagella? No. Using pseudopodia? No. It doesn't have a contractile vacuole also. So it's having two type of nucleus. It's elimination method. Okay. Which one of the following does not is not a eukaryote? Eukaryote means all the other organism except kingdom Monera. Okay. Paramecium belongs to protozoans. Okay. Escherichia coli is a bacteria. Amoeba is again protozoa. Euglena is euglenoids. Okay. So Escherichia coli is a prokaryote because it is a bacteria. Then which of the following is incorrect? Claviceps is a source of many alkaloids and LSD. Okay. Conidia are produced exogenously and ascospores are produced endogenously. Conidia exospores. Okay. Morels and truffles are edible delicacies. Okay. Yeast have filamentous bodies. Is yeast a filamentous body? No. Yeast is unicellular, the only unicellular organism in fungi. Okay. Which of the following statement is incorrect? Prions consist of abnormally folded protein. Is right. Viroids lack a protein coat. Right again. Infective constituent of virus is protein coat. Is it right? No. The infective constituent is a genetic material. So in virus the infective component is genetic material. Okay. Select the wrong statement. Term contagium vibum fluidum was coined by M.W. Beecher-Naik. Is it right? Let's see. Mosaic disease in tobacco and AIDS are caused by viruses. Okay. Right. Viroids was discovered by D.J. Ivanovsky. I said this is not by D.J. Ivanovsky, but it is by T.O. Dyer. So this is the wrong statement. C is the correct answer. Okay. Now which one of the single organism or the pair is correctly assigned? Lichen is symbiotic association. Okay, right. Algae and protozoan. No, it's of algae and fungi. Yeast is used in making bread and beer. It's a fungi. Okay, that's right. Paramecium and Plasmodium belongs to the same kingdom as that of Penicillium. No, Penicillium is a fungi, but Paramecium and Plasmodium are different. Okay. Nostoc and Abina are examples of Protista. Is it right? Nostoc and Abina are examples of kingdom Monera. Okay. So this is wrong. This is wrong. This is also wrong. This is the right one. And answer is option B. Which of the following matches the correct? Mucor reproduction by conjugation of ascomites, agaricus parasitic fungi, basidiomites, alternaria sexual reproduction absent, and which is correct? Okay, sexual reproduction is absent in deuteromites. There is no sexual reproduction in deuteromites. Maximum nutritional diversity, this I said before. Metabolic diversity is in kingdom Monera. Okay, metabolic diversity is in kingdom Monera. Just like that, nutritional diversity is also in kingdom Munir. Okay. Select the wrong statement. Cell wall is present in members of fungi and plantae. Okay, cell wall is there. Pseudopodia are locomotory and feeding structures in sporozoans. Is it right? Let's see. Mushrooms belong to basidiomites. That is right. This is also right. Mitochondria are powerhouse of cells. Okay, that is also right. So, this is the wrong statement. So, option B is right. Mycoplasma can pass through less than 1 micron filter size, it's right. Then mycoplasma are bacteria with cell wall. No, they don't have cell wall. So, statement 1 is right, statement 2 is incorrect. Statement 1 is correct, but statement 2 is incorrect. Okay. And 5 kingdom classification, Chlamydia and Chlorella. This I said it as a PYQ. Chlamydia and Chlorella is belonging to Protista. Okay. Most abundant prokaryotic helpful in humans making curd from milk. Curd from milk is heterotrophic bacteria. Okay. After karyogamy followed by meiosis, spore are produced exogenously. Spore are produced exogenously in, just try to remember, spore are produced in, spores are produced exogenously in these neurospora. Okay, neurospora because it is ascomycetes. Now, just this one too. Halophiles. Halophiles are found in salty area, thermoacidophils in hot springs, methanogens in gut of ruminant animals, and cyanobacteria in aquatic environment. Then you just see the options. Which of the following among the following is not a prokaryote? Not a prokaryote. So, it should not be a bacteria, not a prokaryote. Which is not a Nostoc is a bacteria, Mycobacterium is a bacteria, Saccharomyces is yeast, which means it is a fungi. So, that is the right answer. Which one of the following is not true for fungi? They are heterotrophs, they are true. They are phagotrophs, they lack, which is true. Okay, they are heterotrophs, it is true. Which one of the following is correct statement? Cyanobacteria are group of autotrophic organism classified under kingdom Munira. Cyanobacteria are autotrophic. Okay. Bacteria are exclusively heterotrophic. No, they can be autotrophic also. Slime molds are saprophytic. Under Munira, no, it's under Protista. Mycoplasma have DNA ribosome cell wall. Absolute wrong. So, this is the right answer. Mad cow disease in cattle is caused by abnormally folded protein. Which of the following shows coiled RNA strand and capsomeres? Which shows the coiled RNA strands and capsomeres? Tobacco-mosaic virus. Okay, this was the end of the PYQs. So, let me know the feedback, all your feedback. Just let me know what all I have to do. Should I do more questions or should I do more of the content? Just let me know your feedback. And this was all about biological classification. We will see soon in the next in the class of next chapter. Okay, signing off. Thank you.